I want to talk about Michaelis-Menten and Lineweaver Burk plots. These are two graphical methods that we can use to study enzyme kinetics. Now I want you to remember that many factors can affect an enzyme's reaction rate. Temperature can affect it, pH can, just the amount of enzyme that can affect it can. But in this video we're going to focus on how substrate and how the presence or absence of activators and inhibitors can change an enzyme's kinetics. And that's what these techniques are good for. So Michaelis and Menten, who are pictured in the top row, and then Lineweaver and Burk, who are pictured in the bottom row, develop these graphical methods that we can use in order to do these two things. Let's look at them one by one, beginning with Michaelis-Menten plots. A Michaelis-Menten plot is a scatter plot where we plot the reaction rate as a function of the concentration of substrate. So let me give you an example. There's an enzyme called beta-lactamase. This is something that some bacteria make, and what it can do is it can take penicillin and then hydrolyze it to turn it into something else, penicillic acid. This is something that penicillin-resistant bacteria will express. If they can break down penicillin, then penicillin can't act as a drug against them. It's interesting for doctors who are dealing with infectious disease and antibiotic resistance. Well, here's some data. In order to make a Michaelis-Menten plot, you need to vary the amount of substrate and then determine a rate. And that's what's shown in these two columns. We've got a bunch of different concentrations of penicillin, the substrate, and then the reaction rate. And here's the plot that you would make with them, simply taking each row and treating it as an XY point and then plotting it. So we've got the amount that's hydrolyzed as a function of the concentration of penicillin. And what I want you to see is that the curve increases and then it sort of bends over and it reaches this plateau. Well, curves that show this shape are said to obey Michaelis-Menten kinetics, and what that means is that if we were to fit that curve, we could fit it with the equation V, the reaction rate, is equal to Vmax times the concentration of substrate divided by the concentration of substrate plus Km. And what I want you to notice is that there are two constants here, Vmax and Km, and we want to be able to solve for them. So how do we solve for them? Using the graph. Well, Vmax is the maximum velocity, and you solve for it the way you think you would. I said that when we looked at the graph, we could see the reaction was increasing until it reached some plateau. Well, that highest rate that it reaches, that is the maximum velocity, so that's Vmax. So for this curve, it looks like the Vmax is about 0.62 nanomoles per minute. It's going to be a rate. Km is a little bit more complicated. Km is the concentration of substrate at which the reaction is running at half the maximal rate. So there's a couple steps that we need in order to solve for this. First, we need to know what Vmax is, but we just estimated that, right? I said it was about 0.62. So I know what 1/2 Vmax is. That would be 0.31. And then if I go over to the curve and I find out the concentration of substrate where that happens, which is here, that's my Km. So here it looks like Km is about 4 micromolar penicillin. That's the Km. Michaelis-Menten plots are easy to make and they're easy to interpret, but they can be a little bit inaccurate because being able to calculate Vmax and Km depends on being able to accurately determine Vmax. And you can imagine that we might estimate where Vmax is differently because it's a little hard to tell if it's truly reached that maximal value or not. So when this happens, we might want to use a different graphing method, and that's where Lineweaver and Burk come in. They developed another way to graph data. Lineweaver Burk plots are also scatter plots, but here we plot 1 over the reaction rate as a function of 1 over the substrate. Because we're plotting a reciprocal versus a reciprocal, these are also called double reciprocal plots. So how would we do this? Well, here's the data that I showed you before. But if I want to make a Lineweaver Burk plot, I'd have to pull out my calculator and then take each one of the values for the reaction rate and each one of the values for the substrate and divide it into 1. So for example, at the penicillin concentration of 3 micromolar, well, 1 divided by 3 is 0.33. That's a value. 1 divided by 0.25 is 4. That's where I get my rate, and I'd have to do that for each independent point. Once I do that, I can plot it. And this is what I get. Well, this is a little bit small, so let's blow it up and talk about how we calculate things. Well, the first thing that I want you to notice is that when we make a Lineweaver Burk plot, the data is fit by a straight line. And the equation that fits this straight line is 1 over V is equal to Km over V max times 1 over S, concentration of substrate there, plus 1 over V max. And we can solve then, this is in the form y equals mx plus b, we can solve for the constants V max and Km by looking at the intercepts. The y-intercept is going to be equal to 1 over V max. So if I want to estimate the V max or determine the V max from this Lineweaver Burk plot, I need to go and look at where the line crosses the y-axis. Here it is. It looks like it's about 1.4. And then V max will be 1 over 1.4 or 0.71 nanomoles per minute. So it actually turns out that if you think back to the Michaelis-Menten plot, we were underestimating the V max. Now the x-intercept is equal to minus 1 over Km. So to determine the Km, I'll have to go and look at the x-intercept. Here it is, it's minus 0.2. And then to determine what Km is, I have to take the opposite of 1 over that value, which turns out to be 5 micromolar penicillin. So that's how we'll use a Lineweaver Burk plot in order to calculate these two parameters. All right, well, why would we care? Well, remember how I said that enzyme kinetics are affected by activators and inhibitors? We can distinguish different types of regulators by how they change the Michaelis-Menten and Lineweaver Burk graphs. And I've got a little cheat sheet here showing you the effects of some types of enzyme regulators on Km and Vmax. It's not a comprehensive list. There are some inhibitors that I've left off here, but it's a good start. All right, well, let's try a problem and see if we can figure out what kind of regulator we've got. So in this problem here, someone varied the amount of substrate and then measured the reaction rate either with the enzyme alone or with the enzyme with the regulator. And if we go and we plot that on a Michaelis-Menten plot, then we get these two curves here, right? So the blue is just the enzyme, and then where I've added the regulator, it's the red line. And what I hope you can see is that it looks like the curve is being shifted to the right. So it looks like it's changing the Km. It's a little hard on this graph to see if it's affecting the Vmax because we haven't really reached a plateau yet. So if we wanted to know if it was also changing Vmax, we would have to then go and make a Lineweaver Burk plot. So again, we pull out the calculator, we divide all of these values into one, and then we can plot it. And if we do that, what I want you to notice is now the y-intercept has stayed the same for the two curves, and that means the Vmax of this reaction with the inhibitor, with the regulator, which turns out to be an inhibitor, and without it is the same. But the Km with the inhibitor has shifted. It's moved to the right on this curve, which means it's getting bigger, and that means that this is a competitive inhibitor, because in competitive inhibitors, the Km is increased and the Vmax doesn't change. Let's look at another example. Well, for this one, I pulled data out of a recent paper in which a group was studying something called malic enzyme in a type of parasite called Leishmania. Well, you might be wondering, okay, what's a malic enzyme? So here it is. Malic enzyme is going to be the enzyme that catalyzes the conversion of malate to pyruvate. You should be familiar with malate. It's a 4-carbon compound. We normally think about it as being part of the TCA, but look, here we can take it and convert it into the 3-carbon compound pyruvate. Carbon dioxide will be released here, and this is a redox reaction because we're also transferring electrons to reduce NAD plus to NADH. Okay, so the group here, this research group, wanted to see what happened to this malic enzyme if they added some regulators. So they did what you think they would do. They took the enzyme and they measured the enzyme activity, so the reaction rate, as a function of different concentrations of substrate, malate. They made a Michaelis-Menten plot. Here it is. Now, this particular curve that I'm showing you was done in the absence of aspartate, but then they repeated the experiment, and this time they added 5 millimolar aspartate. And the question is, okay, what effect is aspartic acid, aspartate, having on this enzyme? And what I hope you'll see is this, right? It looks like that when we add aspartate, the reaction rate's increased, and the curve is shifted closer to the origin. So if you look at like the value for 2 millimolar, it's higher when aspartate is present than when it's not present. So it looks like the velocity is higher. And then it also looks like the curve is being shifted to the left, right? Because if you look at the curve with the aspartic acid, it looks like it's pretty much getting to Vmax by the time you get to 1 millimolar malate, whereas for the lower curve, it looks like it's continuing to rise. So based on this, I would think that it's changing both the Vmax, making it bigger, and decreasing the Km. And those would be the two ways that an activator might act. Well, if we want to know for sure, we could make a Lineweaver Burk plot, and that's what they've done here. This is a figure straight out of their paper, and I have to say it's a little bit ugly. We can definitely see the difference in Km here because the x-intercept is different. It's a little hard to see because it's so small if the y-intercept is different or not, but it is. And so this is an activator in which adding aspartic acid will speed up this reaction. And it does it by making it run faster and also making it run at lower concentrations of substrate. Well, I always think it's nice when you have an example to understand why the cell might want to do this. So let me just close by saying, well, why would a cell inhibit malic enzyme if aspartic acid is around? Well, here's a reaction, but this is a different way of looking at the reaction here. So what I've just circled there, that's the same reaction, but now I'm showing it to you in the context of a bigger metabolic pathway. So I hope you recognize the tricarboxylic acid cycle. And remember that although in this reaction we were looking at malate being turned into pyruvate, we normally think about malate being turned into oxaloacetate. So the cell has a choice to make. It has malate and it can decide if it should go and make oxaloacetate or if it should take the malate and make pyruvate. And here's aspartate. And presumably what's happening then is if the cell has plenty of aspartate, it has a way to convert that into oxaloacetate. So it's a way of saying, it's okay, take that malate and turn it into pyruvate, and then you could turn it into something else in the cell. We have a way to make oxaloacetate that we need in order to keep this TCA cycle running. The big picture here is that when you're solving enzyme kinetic problems and you're trying to figure out what a particular regulator does, what it does should make sense in the broader context of the cell. Cells will either speed up or slow down reactions depending on what makes sense to them. And if you are doing an analysis and looking at enzyme kinetics, you want to make sure that the answer that you get makes sense.